Hi, I'm Dr. Joe Chen, and it is my pleasure to give the introduction to my most recently accepted manuscript in fertility and sterility entitled Cryopreservation and Recovery of Human Endometrial Epithelial Cells with High Viability, Purity, and Functional Fidelity. Before I get started, I want to take this opportunity to thank my research mentor, Dr. Giudice, someone with incomparable class and knowledge. I also want to take this opportunity to thank Ms. Jacquelyn Hoffman, my research associate and mentee during the developmental process of this manuscript, and Ms. Kim Chi-Vo, a senior coordinator at the UCSF Endometrial Tissue Bank. I would also like to thank these colleagues and friends for their support throughout this process, as well as for the participation in the beta testing of this protocol, which contributed to the confidence in the transferability and repeatability of this method manuscript. The purpose of this work was to develop an easy, repeatable method for the cryopreservation and recovery of human endometrial epithelial cells. These cells are really important because they essentially form the first line of contact for the implanting embryo in humans. They act as a barrier and potentially as a portal to infections in the upper female reproductive tract, and they can also participate in epithelial mesenchymal transitions. So figuring out a way to effectively cryopreserve these cells can significantly contribute to the reproductive biology and OBGYN community. Now, those of us who have worked with epithelial cells are probably familiar with the fact that they're not as hardy and easy to grow as their mesenchymal counterparts, the endometrial stromal fibroblast. They tend to have limited doublings, they tend to be sensitive to exogenous perturbations, and also they require a matrix such as collagen or matrigel in order to form that strong attachment. So herein, we found that the use of a defined KSFM or keratinocyte serum-free medium with a 1% FBS and 10% DMSO is sufficient to cryopreserve human endometrial epithelial cells and also lead to successful recovery with high viability, purity, and functional fidelity. The reason we use KSFM is because it's well reported by us and others that epithelial cells thrive in this particular kind of medium, so it was logical for us to start there. The other interesting note is that we use 1% FBS as opposed to 10%. The reason is because anytime we included a concentration of FBS greater than 1%, it led to significant fibroblast contamination. We don't have empirical data that can explain this phenomenon, but what we theorize is that there are likely fibroblast carryover from the isolation process that's outlined in the paper, and that because fibroblasts tend to thrive in the presence of a high serum environment, that these carried over fibroblasts, rather than dying out during the recovery process, may be preserved by the high concentration of FBS. And so in general, the technique is very simple. It's very straightforward. Again, I humbly want to restate the fact that we are not claiming to be the first group to do this. Many scientists more experienced than me have done this type of work. We're just inviting people to take this formulation, take these protocols and optimize it, and ultimately contribute to the body of knowledge as it pertains to epithelial cells. And before I close, I just want to take the opportunity to thank the brave women at UCSF who have consented to allow us to use their tissues. These brave women come to UCSF for a benign gynecological procedure, sometimes a serious gynecological procedure, and despite everything that they're going through, they allow us to conduct experiments with their biological samples in the hopes that our research can improve the healthcare of other individuals in the future. If you have any questions, please feel free to contact me or to my mentor, Dr. Giudice. And again, we welcome all criticisms and we welcome all suggestions to continue to optimize this kind of protocol work. Thank you very much.